more than 4 billion years ago, Earth held no cells, no genes, no bodies, only simple compounds, endlessly mixed by heat and water and rock. Most reactions went nowhere. Molecules formed, broke apart, and vanished. But a few arrangements lasted long enough to meet new partners and make something slightly more complex. Chemistry started to show a bias. Certain shapes got copied more often than others. The crucial step came when a molecule did more than survive. It helped make another molecule like itself. That changed everything because copying created lineages. Copies were never perfect, and small mistakes produced variants. Some variants copied faster, some fell apart slower. Now the planet ran a simple test again and again. What persisted, spread. What failed, disappeared. Selection began before life existed. Yet copying alone was not enough. A fragile replicator could not carry much information, and it could not keep that information safe. To build a living body, a system needed a memory that endured, and rules that turned that memory into useful chemistry. It needed a code. That left one central question hanging over this early Earth. How did simple compounds produce a durable genetic script that could write organisms? Then a new kind of molecule appeared, and it changed the rules. It looked like a simple chain, but it carried a sequence, a readable order. That order stored information. And unlike most chemicals, this molecule did not just sit there, it helped reactions happen faster. RNA-like strands acted as both a message and a tool. For the first time, a molecule held instructions and also helped carry them out. When an RNA-like strand made copies of itself, the copying rarely stayed perfect. Small mistakes slipped in. A letter changed, a piece went missing, another piece got added. Most changes made a strand worse at copying, and those lineages faded out. But some changes helped. A strand folded into a shape that copied faster, or resisted breakdown longer, or grabbed building blocks more easily. Information started to matter because it affected survival. Now, the planet held a crowd of competing replicators. Each one borrowed the same raw materials, and each one raced against time before it fell apart. Faster copiers spread. Tougher strands lasted. Better catalysts reshaped the chemistry around them to favor their own reproduction. Success produced more copies, and more copies meant more chances for variation. This was not life yet, but it was evolution in its simplest form: heredity, variation, and competition. RNA had carried two burdens at once. It held instructions, and it helped chemistry happen. That mix worked, but it stayed fragile. Small changes could wreck the message. Copies accumulated damage. Then a quieter improvement appeared. DNA took over the job of storage. It held the same kind of information, but in a sturdier form, built to last longer and survive mistakes. DNA stayed safer because its structure resisted breakdown, and because cells began fixing it when harm occurred. That mattered. A longer, more reliable archive meant more instructions could be kept without collapsing into errors. Life stopped depending on a single molecule to do everything. The system started to separate duties, so each part could be optimized instead of compromised. Now the roles split cleanly. DNA stored the master code and rarely moved. RNA copied small pieces of it, carried those messages, and delivered them where work happened. Proteins became the workers, fast, flexible machines that built structures, moved materials, and sped up reactions. This division of labor scaled life up. With stable memory and specialized tools, complexity no longer hit the same ceiling. DNA could store instructions, and proteins could do the heavy work, but a crucial bridge still had to form between them. Nucleotides and amino acids spoke different languages. Life needed a translator. That translator emerged as a set of rules, a genetic code that matched short RNA words to specific amino acids. Once those matches held, information stopped being abstract. It turned into buildable parts. The engine that enforced those rules came together as the ribosome. It acted like a tiny assembly line, grabbing RNA messages and reading them in fixed steps, 3 letters at a time. Transfer RNAs carried amino acids and fit those 3-letter patterns, like keys finding the right locks. When the fit happened, the ribosome joined amino acids into a growing chain. Sequences finally became proteins reliably. This system did more than make proteins. It standardized meaning. A particular triplet of letters now meant the same amino acid, again and again, across countless descendants. Any lineage that used a different code risked producing broken proteins whenever genes moved or mixed. So one translation system dominated and stayed. The universal genetic code became a deep ancestor embedded in almost all life that followed. On early microbial Earth, copying DNA never stayed perfect. Each split left small mistakes, mutations that sometimes changed nothing and sometimes changed everything. A tweak in one gene could let a microbe handle more heat, tolerate a poison, or grab energy from a new chemical. Most changes failed fast, but the few that helped meant survival, and survival meant more copies of that new recipe. Then evolution gained a shortcut. Microbes did not only wait for lucky errors, they also reshuffled what they already had. Pieces of DNA broke and rejoined in new combinations, like mixing tools from different kits. Recombination produced fresh gene sets in a single step. What seemed impossible for one slow lineage suddenly became reachable because old solutions got rearranged into new ones. The biggest change came when DNA stopped acting like private property. Microbes traded genes sideways through horizontal gene transfer, passing useful sequences to neighbors, even to distant relatives. A trait that appeared once could spread across whole communities. The code became a shared library, enzymes for new foods, defenses against viruses, ways to breathe different chemicals. Life diversified faster because invention traveled. For billions of years, cells stayed small and simple because power stayed limited. Then a rare kind of merger happened. One cell took another cell inside it, not as food, but as a tenant. The captured cell kept running its own chemistry and kept making energy. Over time, the host depended on it. This partnership became permanent. A new type of cell appeared, the first eukaryote. That inner tenant became the mitochondrion, a dedicated power plant. It still carried a trace of its past, a small loop of DNA, separate from the host's main genome. That DNA legacy mattered because it hinted at the mitochondrian's origins as a free-living bacterium. Now, instead of one cell doing everything at once, work got divided. Energy production got outsourced and stabilized. The host cell gained breathing room to grow. With far more energy available per cell, the rules changed. Bigger cells no longer collapsed under their own needs. Genomes expanded because copying and managing more DNA stopped being such a crushing cost. New internal structures appeared and persisted. Compartments, transport systems, and controlled zones for different jobs. Complexity stopped being a fragile experiment and became a workable design. This energy jump opened the door to everything that followed. Once cells carried big genomes and plenty of energy, a new possibility opened. Life no longer had to stay single. Cells stuck together after dividing or gathered in groups, and suddenly cooperation mattered as much as competition. A cluster gained advantages: protection, steady feeding, and the power to grow larger than any lone cell. But size created a problem. If every cell did the same job, the whole group stayed limited. The breakthrough came from control, not new DNA. The same genes stayed inside every cell, but networks of switches decided which genes turned on, when, and where. One cell type ran a different program than its neighbor. Some specialized in movement, others in digestion, others in sensing the world. Division of labor made the group act like one body. A new kind of inheritance appeared, not just traits, but a build plan. With gene regulation guiding development, evolution started experimenting with body layouts. Small genetic changes shifted timing, location, and intensity of gene activity, and the results could look radically different. Tissues formed, then organs, then repeating patterns that shaped whole bodies. This was the doorway to animals, creatures built from many cooperating cells, coordinated by shared DNA and disciplined by gene networks. Complexity stopped being a rare accident and became a direction life kept exploring. In the human line, DNA did more than build a body. It kept notes. Every child inherited a copy that carried tiny changes, some random, some useful, some harmful. Over time, those changes stacked up, like dates stamped into a passport. As groups split, traveled, and settled, their DNA drifted in different directions. The genome stayed a blueprint, but it also became a record of where people came from. When humans moved into new places, old challenges forced new solutions. Cold, heat, thin air, new foods, new diseases, each pushed on the same code. Variants that helped survival spread through families, while others faded. But people did not stay neatly separated. Populations met and mixed, and their DNA mixed with them. Even other hominins entered the story through interbreeding, leaving fragments behind in modern genomes. That meant a person's DNA carried more than one past at once. Some stretches hinted at ancient isolation, others at sudden contact, migration, and blending. Patterns of shared variants revealed kinship between distant groups, and gaps in those patterns marked times of separation. The genome acted like a fossil bed you carried inside you. Not bones in rock, but chemical letters that preserved movement, adaptation, and ancestry across thousands of generations. In the mid-1900s, DNA stopped being an abstract idea and became something scientists could explain. James Watson and Francis Crick proposed a simple shape, a double helix, two strands twisted together. That shape carried a shock. The molecule did not just sit in cells, it copied itself by matching pairs, like letters that always found their partner. Heredity looked less like magic and more like a mechanism. Once the structure made sense, the next question followed fast. What did the sequence say? Researchers learned to break DNA into pieces, sort the pieces, and read the order of its chemical letters. Each new method turned biology into information. A gene became a stretch of text that could be compared, searched, and checked for errors. Disease risk could be tied to a spelling change in the code. Then the scale exploded. In the 1990s, the Human Genome Project set out to read nearly all the DNA in a human cell. It took years, large teams, and massive computing. But it changed the idea of what a genome was. DNA became a reference book for medicine and a record of ancestry. Identity gained a new kind of evidence, a sequence that could be stored, shared, and interpreted. By the 2020s, reading DNA stopped being a rare achievement and became a routine tool. A genome that once took years and huge budgets now got decoded fast, sometimes for the price of a household appliance. That change mattered because DNA reading turned into decision-making. Doctors used sequences to spot hidden risks, match cancer drugs to a tumor's exact mutations, and trace outbreaks by comparing genetic fingerprints. But the bigger shift came when reading the code no longer felt like the end of the story. It became the beginning. With CRISPR, scientists used a guide made of RNA to bring a cutting protein to one chosen spot in DNA, then let the cell repair the break in a new way. It opened paths to fix disease-causing mutations, and it also made it possible to change embryos, meaning edits could pass on. That possibility pulled science into ethics at full speed. A therapy in one person looked like medicine. A change in the germline looked like a choice imposed on future generations. The line between treating illness and selecting traits grew harder to defend, especially when access and power stayed unequal. 4 billion years earlier, chemistry barely managed to copy itself. Now humans deliberately rewrote the code that once arrived by accident.